Hello. Okay, welcome to AP Bio Review video number 3. Let's go with 3. So in this video, we're going to be looking at membranes, cell membranes, and transport, as well as cell communication. So we're kind of going to gloss over all of those topics, and we're going to spend as much time on probably communication as possible, just because communication is generally one of those that is very difficult for a lot of kids. Okay, so let's start off. Let's get into this. So before we talk about cell membranes, we kind of have to talk about this molecule. Now, this molecule is a phospholipid, and phospholipids are the molecules that make up your cell membrane. So your cell membrane is just a bunch of these phospholipids just all sitting next to one another. Now, a phospholipid is made up of two things. It's made up of a phosphate head, and then two fatty acid chains, two lipids. Okay, that's hence the name phospholipid. Now, if you remember back to our biomolecule video, lipids are non-polar. They do not mix with water. Okay, so in order to create a skin around the cell that is that protects it from everything the cell membrane protects it from, while also not mixing with water, we have to create what's called a bilayer, where essentially we have two layers that float on top of one another, where all of the fats are on the inside and all of the phosphates are on the outside. Because remember, the phosphates are hydrophilic, they like water, and they're polar. The inside, the phosphate tails, are hydrophobic, and they do not like water. So being on the inside allows them to not interact with water ever. Okay, so in paint, this is how I kind of drew it right here. So you've got your phospholipids, where our phospholipid tails are on the inside. They are the non-polar side. Okay, and our polar side, our hydrophilic side, is on the outside. Okay. So the whole point of that is to keep water out, and these phosphate heads sort of act as like a protective shield against anything trying to get in. Okay. So that's our phospholipid. Now you'll notice that phospholipids have these unsaturated fats, these kinks, and that's important because that means these phospholipids are not like right on top of one another. There's a little bit of space in between, and that little bit of space is important because it allows certain things to just sneak in between two phospholipids. Okay. That's the difference between a saturated fat and unsaturated fat. Remember, a saturated fat is just a straight fat, and an unsaturated has this bend in it. That bend sort of allows two things to not be able to be right on top of one another. It provides space in between the two phospholipids, and that space is how things can pass through the membrane, small things. Okay, so here we go. You can see this is our phospholipid bilayer. Okay. Notice that they all have these squiggly lines. That's to show you the unsaturated fats. These phosphates can move pretty easily and pretty fluidly. Hence, it's why we call the cell membrane the fluid mosaic model, because these things can move pretty easily. And what makes them take the same shape is the hydrophilic and hydrophobic properties. Okay. We don't want our membrane to be too fluid, because then it's too pervious. It's too easily penetrable. But we also don't want it too viscous, because then nothing can pass through. So both of those are bad. So in order to sort of satisfy both of those things, we have a pretty fluid membrane with a lot of unsaturated fats, but we have cholesterol in there to sort of stiffen it up, to make it a little bit more rigid. Okay. Now, the whole point of the cell membrane is to keep out most things that we do not want in the cell. Okay. But there are tons and tons of things that we need to get into the cell, whether it's nutrients or signals or organelles or proteins or all kinds of crap. Okay. So in order to determine what can get in and what can get out, we determine that based on size and charge. Okay. So we're going to start right. We're going to start right here. So in order to do transports, okay, in order to get things into your cell membrane. So your cell membrane acts as a barrier to keep most things out, okay? Now, certain things are small enough that can squeeze through your cell membrane because, remember, we have these unsaturated fats, or unsaturated fats. So those molecules that are really small, okay, they're just small enough to just slip on by and just pass through the cell membrane really easily. That's what we call simple diffusion, okay? Where a molecule is small enough and non-charged enough to where it can just slide right past your cell membrane to get in really easily, right? So like hydrogen can get in, certain molecules, so long as they're small and they are non-polar, they can go through simple diffusion, okay? But there's a ton of things that are really big or have a charge. So how do we get those things in? Well, we have to use proteins. And so what you're seeing here, these are all of the different kinds of proteins, okay? Now, I did it in this picture in Paint to just because it seemed like the easiest way to deal with this, okay? So I broke it up like this. So there's three kinds of transport that you have to know. There's passive, there's active, and then there's active bulk transport where we're bringing in a lot, okay? So I color coordinated it. So we have passive transport here in green, we have active transport here in blue, and then we have bulk active transport here in this purple color, okay? So we'll start with passive and then we'll just kind of work our way through. All right, so in passive transport, these sticks, these lines here, those are proteins, okay? Now remember, the key to passive transport is there's two properties. Number one is we're always going from high concentration to low concentration, okay? Meaning that if we have a lot of stuff, we're going to be flowing from where there is a lot of stuff to where there is not a lot of stuff, okay? So we're always going to be flowing from high concentration to low concentration. And because of that, we're not going to use any energy. Okay, so that's consistent with all of these different kinds. Now, this first one is what we call a channel protein, okay? And the kind of transport that's going to be done is going to be what's called facilitated diffusion. Okay, so notice that we're still using the same word diffusion, but the word facilitated means it needs help. Okay, it's not simple. It can't just easily pass through by itself. It needs help. So we're diffusing because we're still doing passive transport, but this is going to be facilitated because we need the help of the protein. Basically, these molecules are too big or they have too much of a charge to get through. So this could be something like water. It could be something that has a charge like sodium or potassium or something like that. Okay, anything that has a charge can go through these channels. Okay. Now, these channels a channel like this is just an open channel, so it's essentially like a tunnel on a road. Anything can drive through it so long as it can fit in the actual tunnel. But then we have these things that are called gated channels. And a gated channel, it's kind of hard to tell, but I tried to draw like a lid on the channel, meaning that this isn't open all the time, right? There's a lid on it, and in order to pass through, you have to open the lid. Okay. Now the opening of that lid could be we need a specific response or a specific receptor. We need something to open up this channel. Okay. Now, otherwise it works the exact same way as our facilitated diffusion channel, as our open channel. It just has a gate on top. Okay. We could also have an ion channel, and an ion channel is specifically for ions, so if it has a positive or negative charge, okay? These are all different types of proteins that all do passive transport. It just does them in different ways, right? It's basically just ensuring that we're only letting in certain molecules, right? The ion channel is only letting in molecules that have a charge. I try to draw a little square space for these little square ions because if it doesn't fit in the protein, remember proteins have a very specific shape, and if those ions don't fit in the specific shape, they're not going to pass through. So the point of the gated channel and the ion channels is that we're only letting in the things that we want to let in, okay? So that's what we're seeing here, right? We have these protein channels, we have gated channels. These are much better drawings than the crap I did in Paint, but you get the idea. And then we have these transport proteins. These are the ion channels I was talking about, that the thing has to fit into the protein before it can actually be transported in. So these proteins are sort of acting like bouncers at the club, right? They're keeping out the things that they don't want out and letting in the things that they do want in. They're being selective, right? Because remember, the whole membrane is selectively permeable. Now, if we continue on, we'll go to active transport. Now, the main difference between active and passive transport is that active transport is going from low concentration to high concentration. So we're going the opposite direction. So here we can see we have only 3 blue dots to our a lot more blue dots. We're going from low concentration to high concentration. And these require energy. So the main kind of protein that we're going to be using for active transport is called a pump, okay? Where in order to move these things from low to high, it takes a lot of energy to keep the high concentration on one side and the low concentration on the other side. So, because remember, this high concentration, they're trying to get back in. They want to naturally equalize out, okay? I said these proteins are bouncers at a club. If nobody's in the club and everybody's on the outside, they're all going to try to bust in. And it's going to require that bouncer a lot of energy to keep those people out. So in order to maintain that, this thing has to use energy to get the people out, or to get the molecules out that it doesn't want in. So that's where this ATP is going to come in. So we're going to use the ATP to pump out these molecules from our low concentration to our high concentration. Okay, so we're going to be using energy with active transport. If you had a boat and the boat was filling with water and you were trying to pump it out, you'd either have to shovel it out with a bucket or something, which would require energy, or you'd need an actual mechanical pump, which you'd have to plug in, requires electricity, which is energy. Okay, so pumps require energy. Okay, now these guys down here, these are the bulk active transport. So these are still active transport. We're still going from low to high. We're still requiring energy, but these proteins, both passive and active, these are like if you're trying to get in like one thing at a time, right? There's not really there's no sense of urgency. But if your body is like, no, we need to get a lot of this thing right now, that's where this comes in. Okay. Now there's 2 kinds of bulk active transport. We have endocytosis and we have exocytosis. Endocytosis is when you're trying to get things into the cell. Okay. So in this picture, the bottom part of the screen is the in, and the top part is the out. Okay, so the endocytosis is when you have when you're coming into the cell. Exocytosis is when you are going out of the cell. Okay, now what you'll notice here is that we're using our cell membrane as to create a bubble called a vesicle. Okay, so we're actually having to consume part of our cell membrane to create this bubble for these different molecules to float in. Now that's cool because anytime the cell receives a vesicle from somewhere else, it's going to just sort of rejoin up with the rest of that cell membrane. So we're not necessarily losing any cell membrane as we're doing this. Okay. Now I've got the 2 types of endocytosis down here. We have pinocytosis and phagocytosis. Just to simplify it, phagocytosis is for like solids, like food. Pinocytosis is for liquids, so like water. So like if you drink a lot of water, your body wants to distribute that water to every cell as quickly as possible. So it's going to be using endocytosis, it's going to be using pinocytosis to get large amounts of water to as many cells as it possibly can, as quickly as it can. We don't want to get water 1 molecule at a time. You need water now. So we get in as much as we can, as quickly as we can, using endocytosis. Okay. Exocytosis is the exact same thing. It's just going out. Now, this one in the middle, this is through receptors, because remember, we talked about receptor endo and exocytosis, where these Y-shaped things, those are receptors. The advantage to this would be so let's say we have 2 types of molecules. So we have these circles and we have these dots. If we wanted to get in more circles than we did dots, that's our target molecule, these circles will bind to the receptors, and this vesicle will only be made once these receptors are bound and activated. So the idea is that you're getting more of the target molecule that you want in, and not a bunch of crap that you don't want in, right? Because these other 2, these are just essentially holes, and as soon as it fills up with something, it just comes in. So it could be a lot of, if it's water, it could be a lot of water, it could be a lot of not water. But with the receptors, you're getting more of the target, whatever you want to, whatever you're trying to target. Okay. So those are all the types of transport in a very quick nutshell kind of explanation. So these are the receptors I was talking about. A more well drawn picture of the receptors. So So we have to bind to the receptors in order to create the vesicle, and that's when our vesicle gets made. Here we have an example of exocytosis, okay? And you can kind of see how the vesicle is going to rejoin with the cell membrane, so we don't really lose or gain anything from our cell membrane. Are there any other pictures I want to show? I don't think so. That pretty much covers most transports, okay? And generally speaking, in the past, when they've asked transport questions, it's been about like not even differentiating active and passive. It's been just like, describe a specific type of protein that is used in passive transport, or describe a protein that's used in active transport. So I would highly recommend that you know these types of proteins and the differences between them. What's the difference between a channel protein and a gated channel protein, or a transport protein, or that kind of stuff. Now, I didn't talk about the receptor protein, and I didn't talk about the glycoprotein, okay? And the reason is because those guys really aren't used in transports. They're proteins that are bound within the cell membrane, but these were all the ones that are used in transport, okay? The other proteins that we have in our cell membrane are going to be these guys. So this is where our receptor protein comes in, and our glycoprotein and everything else. Okay, so if we look at this picture, so we have our transport proteins, right? They're either actively or passively letting stuff into and out of our cell. That's the point of those proteins. We also can have proteins that are in the cell membrane that don't do transport, but they have other functions. Okay, so for example, we could have an enzymatic protein where it's an enzyme, because remember, enzymes are proteins. It's just bound to the membrane. It can't freely float throughout the cell. It's just specifically bound to the membrane. We have signal proteins, receptors, which we're going to talk about here in a second. We can have intercellular joining proteins. I call them anchors because that's what they do. Cell-to-cell recognition proteins, that's our glycoproteins. So our glycoproteins here, they have carbohydrates attached to the protein. And the way that one cell knows that another cell is two cells both know that they're your cells, is that they have the same fingerprints. And those fingerprints in this case are these glycoproteins. So when two cells are butt up against one another, they check the carbohydrates and the glycoproteins to make sure that they are your glycoproteins. If it's not your glycoproteins, your body thinks that that's not your cell, and it will try to kill it. And then we have these anchor proteins that bind to the cytoskeleton. Okay, so remember, you have the cytoskeleton, which is just a bunch of filaments that just connect and hold all your cells together as best they can, because cells are really fluid. And so we have proteins to sort of anchor those cells to the cytoskeleton. Okay. Now let's talk about receptor proteins and enzyme proteins and cell-to-cell communication. How do cells talk to one another? Okay, because transport and the cell membrane kind of go hand in hand with that. And so the best way to describe it is, so I've got a bunch of different proteins here. And we'll talk about what each one does, okay? But the main thing that you got to know is this, okay? Now, we talked about this a lot in class. This is what is called the signal transduction pathway. This is essentially a simplified version of how most cells go through cell communication, okay? So there's a couple different parts for all this, and this is kind of what all of these different pathways have in common. And then I'll get into the individual differences, but this is what all of them have in common, okay? All of them have a signal, okay? And that signal is what we call a ligand, okay? The ligand could be anything. It could be a hormone. It could be something your body produces. It could be food that you eat. It could be a nutrient. It could be anything, okay? But it is something that is floating within your body that then binds to a specific receptor, and that receptor activates a response, okay? That's called a ligand. Now, once that ligand binds to the receptor, that leads to our second step, which is called our reception, okay? Now, so you'll notice that this ligand is very specific in terms of its shape for this receptor. It's got the same sort of, triangular shape here. That signal is going to bind to the receptor, and it has to bind specifically to that receptor. So if this thing tries to go in a different receptor with another shape, ain't gonna work, okay? It has to fit into that receptor. So that's where we get our reception. Now, the reception is going to activate that protein. And in turn, that protein is then going to activate other proteins. So you can kind of see here, I drew it as this protein activates these 4 other green proteins. And then those 4 proteins will then go on and activate 4 more, right? Or 2 more. And then those will go and activate more. And so we get this amplification of our response where, if you think about it, if you get a signal, if you get something really important and you want to tell everybody, you don't just go around one person to one person. Hey, by the way, did you know this? Hey, did you know this? Hey, did you know this? No, you're going to get on social media and you're going to blast it out. You're going to get a megaphone and yell it, right? You're going to try to tell as many people as quickly as possible. That's what the amplification is. It's trying to spread the word of that signal as quickly and as efficiently as possible. And so we get an amplification of the signal from the receptor, okay? We don't just send out, it's not just one protein activates another protein, activates another protein. It's one protein activates 4, and then each one of those activate 4 more, and then each one of those activates 4 more, and we just get this big, long amplification, okay? That step is called transduction, okay? Where this group of proteins will activate this group of proteins, will activate this group of proteins, okay? And then our last step is our response, okay? Now, if they ask you a signal transduction question, you can't just say, hey, and there's a response, okay? You have to specifically say what response is there, what kind of response could it be, okay? And most of the time, the best answer that you can put there, the easiest, most vague answer while still not being vague, is that it either activates or deactivates a gene, okay? So for example, you can see here we have our protein that when it's activated, so let's draw one. Let's, because we're doing this in green. So let's say we have this molecule, which if you remember is called our ligand. I'm just going to call it our lig. Now, our ligand is our signal. That's our step 1. It's going to fit in to our receptor, which is step 2. That's our reception. So this is going to fit into this thing. Now, this protein is then going to activate it's going to activate a protein, which is then going to activate more proteins, which is then going to activate more proteins, until eventually we get to a point where the last protein in our transduction is going to go into our nucleus. This is my poorly drawn nucleus, and then it's going to find a specific gene, a specific section of DNA, and either activate it or deactivate it. Okay, that would be an example of a response. Now, there's a ton more responses that could be activated. This is just a good example of a response that you could put. Okay, you might get a question. It's like identify an example of a response in signal transduction. Turning on or off a gene is a good example of a response. Okay. Now this is the signal transduction pathway. This is, again, these are all things that all of these different specific pathways have in common. Some of them vary in how they do it. Okay, so specifically we talked about. Let's do this one first. So this example where our receptor is bound within our membrane. It's also possible that we have a receptor that's not bound within our membrane. That receptor is instead floating within our cytoplasm, okay? Which is fine, except now our ligand, I'm just going to draw this as an L. That's cool, but the ligand has to be small enough to where it can pass through the cell membrane, or it has to go through one of the channel proteins that we just talked about, okay? So in order for this signal to work, it has to pass through the cell membrane. That's just an extra hurdle that this ligand has to go through. So it's a little bit easier to have your receptor protein bound to the outside because this ligand, it doesn't have to attempt to go through the cell membrane and then get, rebuffed and then it has to figure out what to do. No, it can just bind without having to go through the cell membrane. This little guy has to go through the cell membrane in order to get in, and that could be a problem. But this is an example of a receptor that is intracellular. It's inside the cell. It's not bound in the cell membrane. Okay. But otherwise, it works the same way, right? We have the signal, it binds. We have a transduction, which is this arrow, and then we get a response where we activate a gene. Okay. We also have an ion channel where our receptor. So we have our ligand here. It binds to our receptor protein, and then there's no transduction. There's no response. Well, the response is that this protein. I guess the transduction would be this protein activates, and then the response is the channel is now open for all of these green little dots. Who doesn't like green little dots? Now can come in. Okay, and once that job is complete, the ligand detaches and the protein is deactivated again. Okay, so this is sort of an example where the signal transduction pathway is just all contained within one singular protein. Okay, now here we have a TKR. Okay, I drew it right here. The TKR stands for tyrosine kinase receptor. Okay, now what's happening here is you've got your signal, and that signal basically causes the protein to change its shape. Okay, so these are two proteins that are basically an enzyme. Okay, now when the signal binds to our receptor, those receptors shift and connect together. Okay, and the response is these guys now acting as an enzyme. So I drew it right here. This is a little bit easier to understand, I think. So you've got your ligand. The ligand will attach to the receptor. Okay. And when the ligand attaches to the receptor, it causes our enzyme to change shape. So here I drew it as a square. Maybe now, once the ligand binds, now it's no longer a square. Now it changes shape to, I don't know, let's say it changes shape to a circle. So now this enzyme has a different active site. So now maybe all of these substrates, that's a substrate, they couldn't bind before because, remember, we had a square shape. But now that we have changed our active site to a more circle shape, these guys can bind no problem, and this enzyme can do its job. Okay, so our signal was our ligand. We bind to our receptor. Our transduction is this protein changing its shape, and then the response is that this enzyme now functions as an enzyme. It now can do whatever it's trying to do the reaction to this substrate. Okay. If you remember, this is what's called an allosteric enzyme, where the active site changes shape. Okay. But these are the same things. This picture is a little bit more confusing. I think this one is a little bit more, I don't know, streamlined, easier to understand. Simple. That's the word. The next one we have is our G protein. Okay. Now the G protein is where instead of the receptor and the enzyme all being the same protein, they're two separate proteins. So here we have our receptor. So we have our ligand that binds to our receptor. But instead of the enzyme just switching shape, we have to activate something in order to activate our enzyme. And that is what is called the G protein. That's this green thing right here. So when the ligand binds to the receptor, it's going to activate the G protein, and the G protein is going to slide over and touch the enzyme. And when the G protein touches the enzyme, it's going to turn this enzyme on and do the exact same thing that happened over here. It's going to change the active site of this enzyme from being a square to now a circle, and now this enzyme is active and it can bind to our substrate. Okay, so we can see here this is our receptor protein. Our signal binds to it. Our G protein activates and is going to slide over to then activate our enzyme. And when the enzyme is activated, our cellular response is the enzyme, does whatever reaction the enzyme's trying to do. And then once it's completed, the G protein detaches and it slides back over to our receptor, and this enzyme goes back to being off. Okay, so this enzyme is only active when this G protein is attached to it. Okay, that's the way to control when this enzyme is on and when it's off. This enzyme is only on when it receives this original signal. Okay. So maybe let's say this signal is lactose. Your body eats lactose. The lactose binds to the receptor, slides the G protein over. The enzyme is now active, and this enzyme is now breaking down lactose. Okay. As soon as the lactose is broken down, there goes our signal. Our signal's not there anymore. We deactivate our G protein because our signal's gone, and this enzyme goes back to being turned off. Okay. So, what all three of these have in common so far is that we can turn them on and turn them off only when we need them, depending on if the signal's there. For all of these to work, the ligand has to be bound to the receptor. If the ligand isn't there, if the ligand gets removed or it's get consumed or whatever, these receptors don't work, which means none of their transductions or responses do anything. Okay? All right, so we've got the anchor protein here, and this is our glycoprotein. I'm not going to write that because I'm just not. That's a glycoprotein. Just know this thing right here, that box, glycoprotein. Okay. there's already too much of my crappy handwriting all over this thing for you to be subjected to more of it. So that's a glycoprotein. All right. Now, this will be the last thing. This is different types of signal transduction. Okay. So we've got our ligand, our receptor, we have our transduction, and then our response. Okay. We already walked through that pathway. Okay. Now it's possible that we could have another different pathway, and that is a pathway of inhibition. So you can see right here, this is our general normal signal transduction pathway. Here, the second picture, you can get a little bit better idea of the amplification. So we have our ligand, it binds to a receptor. The receptor then activates this spherical protein, which then can activate two different proteins, so we can get multiple different responses. Okay. Now here, this third picture is where we can either get activation or inhibition. So for example, instead of maybe one signal, maybe we need two signals. So we need this triangular signal and we need this spherical signal. And the response only happens when we get both signals. Okay. It could also be that this circle ligand is an inhibitor, meaning it prevents this pathway. Okay. So that's what we drew here. So let's say this ligand binds and it's doing its normal signal transduction pathway. Okay. But then we get a second ligand, which we're going to do in red. So we're going to get this second ligand that's going to bind to this receptor. Okay. Now, originally we had this receptor that is activating our circle protein, which then activates our triangle. That's a triangle. It activates our triangle protein, which then activates our square protein. Receptor number 2, I should do that in red. Receptor number 2, when the ligand binds to receptor number 2, it creates the star protein. It activates the star protein. And let's say the star protein is going to inhibit the triangle. It's going to inhibit this triangle, which I am drawing with this little, not with arrows, but with this little, this little shield blocking thing. Now, what this means is that this pathway is going to continue to go until this square ligand is introduced. Once the square ligand is introduced, it's going to bind to our receptor, which activates the star protein, and the star protein is going to come over here and inhibit this triangle. If the triangle is inhibited, that means it can't activate the square. If the square is inactive, that means it can't turn on or off this gene. So all of this crap doesn't happen after the star protein inhibits the triangle. It stops right there. That's how inhibition works. So maybe this pathway continues to go on until this ligand gets introduced, and then we stop. Okay. To give you an example, let's say, when you're a kid, you're 10 years old, right? And you're growing, and let's say, okay, it's time for you to grow, right? And so we activate this ligand, okay, and we grow, okay? But in order to grow, you need certain things, right? You have to have a certain amount of nutrients, you have to have a certain amount of food, you have to have a certain amount of calcium for your bones, right? Maybe you're nutrient deficient and you don't have those nutrients in order to grow. So this ligand is going to inhibit this pathway until those requirements are met. And once those requirements are met, it will detach and we'll let the pathway grow we'll let the pathway go so that you can grow. Yeah. So that's how inhibition works in this signal transduction pathway. Okay. Here we get a little bit better view of amplification. Okay, so same steps. Remember, we have our signal, we have our receptor, and then we have transduction, where we take an inactive protein, we activate it, which then activates another protein, which then activates another one, and then we get that response. And again, in most of these pictures, the response is always we activate a gene or we deactivate a gene. We take an active gene and turn it off, meaning we're not making those proteins anymore, or we take an inactive gene and turn it on, which means we are activating those proteins. Okay. And that can lead to really crazy, really scary looking pictures like this. Okay. Now, I've said before that they're not going to give you anything this insane. I just like these just to freak you out. But what they have done is they have given you a picture like this, right? It's not labeled. It's just, this is the picture that they give you. Okay. I think in the 2013 test, this was the exact picture that they gave. Okay. It wasn't labeled. It was just this whole thing. Okay. And one of the questions was label this picture. And you had to go through and say there was a box here, a box here, and a box here. And you had to label, okay, this is reception, this is transduction, this is the response. That was a point. Okay. So, and then there was a follow-up question of like, imagine this protein gets inhibited. What happens? And you had to say, oh, well, if this protein gets inhibited, that means that this protein doesn't active, which means that the cellular response, whatever that is, doesn't happen. And then the third question was, well, provide an example of a proper response. And you could have said a proper response would be the activation of a gene or the deactivation of a gene. And so if this protein inhibits this protein, that means that that gene doesn't get active or it doesn't get deactive. Okay. So they're not going to give you something like that. But I have seen them give literally this exact picture before, and some of these other ones. Okay. So, they might ask you to label a G protein. Well, not on this one, but they might show you a picture and they might use it as context. Okay. More importantly, they're probably going to give you an example of maybe an experiment that's done involving G proteins, or an experiment that's done involving signal transduction, or an experiment that involves, you give a patient this drug, which is the signal, and it has this response. And then if we give a patient this other drug, the circle drug, it get this response. How could that affect? What effect could that be having on the patient? You could say, well, this drug might be the activator for response number 1, but this drug might be the activator for response number 2, or this drug might be the inhibitor for this drug, right? Something like that. That should pretty much cover it. That is a very very quick and broad covering of cell membranes and transport and cell communication. Okay. I think after this video, you're going to be doing we just did free response number 4, or we just did free response number 3. So you should be doing free response number 4. Okay. The next video will be covering, I think, cell cycle cancer and maybe transcription translation. That's kind of a lot. We might do cell cycle and cancer, and then I might do a second video for transcription translation. But yeah, I'll see you guys in the next one.